Hi everyone, I'm Hazel. Welcome to this week SCSE Club. So for this week, we will show you, as you can see on the screen, we'll show you a quick guide of how to handle GeoMx data on SpatialX, including data submission, how to combine multiple samples with batch correction, perform the gene expression analysis, the ROIs annotations, and DG analysis. And at the end of this presentation, I will also show you some of different methods to visualize our data on BioVinci. So again, I'm Hazel from the Biotrain team, and it's very nice to meet you all today. Please feel free to let me know if you have any question and anything that you would like to comment. You can always put into the Q&A box or put into the chat. So we will try to answer all the questions that you may have. Okay, so now let me go ahead with the quick introduction of the agenda today. So as I just mentioned earlier, we'll go through the best practice of how to handle GeoMx data, from submit the data, combine the data, and perform some of the downstream analysis and visualize the data as well. Okay. So before we dive into the data itself, I would like to show you like a quick overview of the spatial transcriptomics technology. Okay, so as you may know earlier, our SpatialX is our platform to analyze different spatial transcriptomics technologies. And so for the spatial transcriptomics technologies, they usually fall into the two main categories, including imaging-based and sequencing-based. On the left side here, we have the imaging-based methods such as xenium, merscopes, and cosmic. So these technologies rely on highly multiplex fluorescent probes. As you can see here, different kind of probes that physically bind to RNA transcripts within the tissue slice, and then the location of these transcripts is then captured and sequenced by taking multiple high resolution images. And on the left side, we have, oh, I'm sorry, on the right side, we have the sequencing-based methods, which include like Visium, Visium HD, Stereo 6, and GeoMx. And these methods use a different approach. Tissue is placed directly on a slide containing a grid, of spatial barcode, and then the RNA is captured, reversely transcribed, and tagged with these barcodes. Then the whole library is sent out for traditional sequencing. So yes, the main technology that we are looking at today is GeoMx. The full name is the GeoMx Digital Spatial Profiler, DSP. And so GeoMx is a sequencing-based spatial transcriptomics technology with unique regions of interest. And one of the very big advantages, a very huge advantage of GeoMx is the ability to capture a large number of genes. I would say like a whole transcriptomes of around 18 to 19,000 genes with a very nice sequencing depth. And then within GeoMx, even though you don't have the single cell resolution, you can still select the exact ROI that you want to work with. You can even select like a whole region of ROIs containing from 50 to 100 cells, or you can even base on the fluorescent staining to pick the ROIs based on different protein channels. We'll want to see the data a little bit later. Okay. So here first, we'll see first of, so how can you submit the GeoMx data onto SpatialX? Okay, so to submit the data, like the GeoMx data onto SpatialX, you only need two files. First, you will need the image file in the OMATIF format, and you will need the biological probe QC in the Excel format. And I understand that you guys usually have multiple samples with like multiple images captured differently. And if you want to submit these multiple samples onto SpatialX, you just need to submit multiple OMATIF images along with one same biological probe QC since this single biological probe QC file contains the metadata and also the expression matrix for all the samples within your data. Okay, I will go quickly to SpatialX to show you how you can navigate to the data submissions. Here, we're in the homepage of SpatialX. You can go to New Study button in here, the blue button. I will make it a little bit bigger. And after that, you can input the name. You can also change the species accordingly to your data. And when you go next here, you can add new data right there. And in order to submit the GeoMx data, you can select the platform of GeoMx and just drag and drop the images and also the biological probe QC file onto the submission panel, submission field in here. Okay, so we'll go a little bit back to the root, back to the machine itself. So how can you get the files from the GeoMx DSP machine? So in here, we already capture, we'll give you some of the screenshots of the UI on GeoMx DSP, where you can export, as you can see on the right side here, you can export one probe QC file, which will be later on biological probe QC Excel file. And for the image, you can export in omitted format for multiple images that you may have. Okay, and in here we go next, and just another like a little bit detail on how the file should look like, how the Excel files, because this is like a very popular question that we usually receive. So as you can see in here, the biological probe QC file structure usually contain 5 sheets like this one. Segment properties, target count matrix, bioprobe properties, and the dataset summary. I actually have the actual file here for your okay, let me here. I have the actual file for your reference. So as you can see that the first sheet in here, which called segment properties, this will contain the metadata information of all samples. This data set that I am having here is actually the example data set directly from GeoMx. You can try to search for GeoMx example dataset or GeoMx example human brain dataset, and you will easily find on the GeoMx website. Okay, so in here you will see that I have the sample number 1, and I also have sample number 3. So all the samples metadata will be stored within this segment properties sheet in here. Okay, let me go back here. And the next worksheet that you might need to notice is the target count matrix, because this target count matrix is the actual expression matrix of your data, which contain the like the ROIs by gene matrix, where each column in here represents one ROI and each row, okay, each row represent a gene. So there's a very interesting fact about the GeoMx expression matrix is that within this biological probe QC file, all the genes that appear in this matrix always have an expression level higher than 0. So all the genes in the matrix will actually express in all the ROIs. So I can go back to the sheet, for example. So as you can see, the smallest number you can find in this sheet will be number 1. So this is the minimum count for every gene in the GeoMx dataset. You can also take a look at the dataset summary sheet, which contain some of the QC params applicable and apply for your own dataset. So you can take a look at this to understand better of how the data was QC and how the different thresholds was applied on the GeoMx DSP machine. Okay, now we go back to the slide here. So okay, now this is the next step of data submission. Okay, as you can see in here, when you open the GeoMx submission panel, you will see the panel in here. You can just drop and drag the file. However, how to actually drop and drag the file and how to actually submit and upload the data to SpatialX. So you have two ways. You have two ways to perform that. You can either submit the data from S3 bucket, and this is actually recommended just because it maintains like a better internet connection and a more stable connection for your submission. And so in order to submit the data from S3, you can link your bucket to SpatialX, and then you can just open My Data. You will see a cloud button. Actually, not just S3. We also support the even Google Cloud if you do have. And after link to SpatialX, it will appear here under cloud icon. And you can just open the bucket and open the dataset and drag and drop the file, the corresponding file, onto this submission panel. Okay. So, however, there's another way. There's another way that if you already store your data on like a local computer, you can also upload it directly from your local computer. So in order to do that, you will need to navigate to the upload panel in here, upload tab. Let me go to the platform. When you click on the upload tab under your storage in here, you will see an icon to actually drag your file from your local computer to SpatialX. And after the files are uploaded onto SpatialX, you can then go back to My Data under the upload folder. So there are two upload. The upload tab to upload the data and the upload folder under My Data to actually submit the data onto SpatialX. Okay, and this is how you can go. If you have multiple samples, you can just, add new sample to submit it along the way. Okay, now let me go back to the slide. Okay. So that is in here. Okay, that's how you can upload and submit your data onto SpatialX. And now, once you already have the data submitted, how it will look like. Let me go next to SpatialX in here. So this is how your data will look like after the data submission. You open your data set from the SpatialX home screen, and this is what you will see. This is what you will see. You will see your image with the different ROI, and in the main panel in the between here. On the right side of the screen, you will see the gene panel to query gene, and also the metadata to query for different metadata information from the Excel file. So in order to adjust the visualization of different components on, the image or like the ROI or like the the gene expression, you can navigate to the left panel, the lower left panel of the Preferences tab. In here, when you open the Preferences tab, the first thing you will see is the Image tab. Within the Image tab, you can see and you can also adjust the appearance of different protein channel. Like this one, I can turn it on and turn it off, like this one. And I can also click on this Configure Channel button to adjust the actual signal of different channel, and it will apply directly on the image. And if you want to adjust like the appearance of different ROIs, you can navigate to the label tab right there, and we can increase or like decrease the opacity. Like in here, we can zoom in, like this one. You can increase or decrease to, to have it visualize the image in the background. But you can turn it on and turn it off as well. Okay, so the first question that we're in here is that how about if you have multiple samples and you want to combine them together and to also remove the batch correction between different sample, how can you do it? Okay, so in order to combine the data, you will see that on the main screen here, there's a little drop down button right here. Okay, when you click in this drop down button, you will see your individual sample, right? In here, I have 3 samples of human brain of number 1, 2, and 3. In order to combine these samples together, navigate to the Combine Data button up here and click on it. So I can put this as a Combine one. And for the technology, you can select the nanostring GeoMx and select all the sample to combine. In here, there's a quite important field to notice is the metadata handling. This is how we handle the metadata from individual samples when we combine it all together. If you select ignore, then all the metadata from individual sample will be ignored and discarded from the combined sample. One of the common practice is select combine, because selecting combine will combine all the metadata in different or like in individual samples together, and metadata with same name will be merged into one. So for example, if you have like the segment labels like this one in here of a different cell type, you can merge them into one single metadata, or you can also select separate where all the metadata from individual samples will still be there. However, they will stand separately with different names so that you can easily distinguish between metadata from sample 1 versus the metadata from the sample 2. Okay, and you can see in here, you can select what kind of images you want to combine as well. And when everything is complete, you can just click combine here. And we go directly to this tab where I already combined the data. And this is how it will look like. The 3 samples will now be combined together all together in here. Okay, well, that's how to combine the data with metadata. So how about the batch correction? How about the batch correction? So when you combine you combine the sample, they will automatically batch corrected using SCVI. How can you know that? Okay, now we go back to SpatialX. Here we have different ROIs. Oh, there is one notice. All the cells, the term cells in SpatialX refer to, specifically for GeoMx, refer to the ROIs. So when you see 173 cells in this combined data, it means there are 173 ROIs in this combined data. Okay, now we go back to the batch correction. In order to see the UMAP, you can go to the 2D mode in here. Okay, and it will show you directly. Now you can select the metadata and I can have the batches information. Okay, which stand for 3 different samples, and you can see they are quite like blend in all together. I can also go to the preferences to make my point size a little bit bigger, like this one. Okay, like that one. Okay, you can even turn on the X and Y axis of the UMAP using this button. So you can, this can be easier for you to export the figure like this one. You can export the actual figure with the legend or with the axis information. Okay. Okay. So in here, I want to highlight again that when you combine the data, we automatically perform batch corrected using using SCVI, and the default batch correction use samples, different samples, like in the default batches information as the batch key. And you can always rerun the batch correction. You can always rerun using a different batch key. In here, when you go to the 2D mode, you will have the embedding button on the upper left panel. When you click on it, okay, you can run new PCA or like new SCVI with or without batch correction. Okay. Now we go back here. Okay. So here is how you can rerun the embedding. There's a little suggestion for rerun the embedding. So we usually for GeoMx data, we recommend using raw count or log normalize, for the best result, just because we noticed that within the in here, within the different normalization method, we also have log 1p. However, the log 1p usually eliminate the differences between. So based on our experiment, it's usually eliminate the differences between the different ROIs. So I would say that using the raw counts or the log normalized is a two good options to go with when you want to perform, a new embedding. Okay. Okay. So another question, another very common question is, so how about if you want to rerun the embedding, but you want to rerun it after removing the control ROI? In the GeoMx data, there is a popular practice that you have one control ROI that you want to remove it from the final embedding. So in order to do that, it's quite simple. You can just hold and select all other ROIs except for the control, and then you can just select the subcluster. Okay, after select the subcluster, you can save the subcluster. And now the new subcluster only contain all ROIs except for the control. Then you can rerun the embedding and clustering using this new population. Okay, in here, I just want to show you here we can open the metadata of the here ROI label, for example. Okay, so if you want to select multiple, you click on the text, not the checkbox in here, right? You click on the text, and then you can hold the shift button to select all other samples like that, except the ROI number one. Let's hypothesize that number one is the control. Then you can subcluster them, and after subcluster, you can save it, and it will appear up here. So this is one example that we performed earlier, and you can, rerun the embedding that way. Okay. Oh, here, in here. So after rerunning the embedding, of course, you will want to rerun the clustering as well. So how to rerun the clustering? You just simple select the clustering in here. After we run the embedding, you can directly run the Disney UMAP or rerun the clustering. The clustering button will be here on the upper left panel. It's very straightforward. However, there's one notice. There's one very important notice is that for the number of neighbors by default is set for 90, which is suitable for single cell spatial data. However, for GeoMx, usually the data set will have small number of ROIs, like around 60, 40, or whatever. So you will want to consider adjust this number of neighbors, right? In here, you can reduce the number of neighbors to equal or smaller than your total ROIs count. Because if you have less than 90 ROIs, running clustering with the number of neighbors of 90 will return an error. So yes, this is one notice that you should consider adjust when you play around with the clustering feature. Okay. Okay, so next, after you have the different clustering, the Louvain clustering, and the embedding is ready, now you can finally perform the gene queries and perform some manual ROIs annotations. Okay, first, how to query for a gene. Okay, now we go back here. We'll go back to the image mode. Okay, I will turn this off. Okay, I will bring it up here, zoom it in a little bit. So, for example, if you want to query for a gene, you can just go to the gene panel on the upper right side of the screen and just put any gene of interest, apply it here. Okay, and that should be fine. Okay, and now you can also input more genes to query for multiple genes at a time. Or in here, all means query for multiple genes at a time, right? You apply it. And so in here, when you query multiple genes, there is a very interesting case. When you query for multiple genes, we have the co-expression to represent for ROIs that express gene 1 versus ROI express gene 2, and ROIs that express both of the genes. However, due to the nature of GeoMx data, where all the genes are expressed, the co-expression should not, is not really helpful in this case, because you can see all ROIs co-express the two genes, right? So in that case, we recommend you to refer to AU cell score. So AU cell score will calculate the enrichment level of your query genes in individual ROIs, and that should be like a better way to evaluate the co-expression level of multiple multiple genes at a time. In here, all genes are expressed. So for that reason, you can try to use the AU cell score. Okay, and after you select different genes based on the AU cell, or like select different ROIs based on the AU cell enrichment score, you can annotate those ROIs based on the AU cell score. Just simple, drag a long distribution heat map in here like that, or you can fine tune the selection range. I will turn it into 0.4, enter, and you will see there are 28 cells selected. You can annotate these ROIs into, like a new metadata like that, and new metadata will appear under the metadata tab. So that is one way for you to query for genes and to annotate different ROIs as the different name. Okay. Okay. Now, next. So of course, querying for the genes and also manually annotate the ROIs is one option. Another option is you can perform the macro genes. So of course, you have in here. Let me go here. You have the Leuven clustering, right? You have the Leuven clustering, and you can base on the Leuven clustering to find the macro genes of different ROIs. For example, you can click on one cluster like this one, and you can select macro genes. So in here, a very important notice is for macro genes and also DGE, because macro gene is just like a simple version of DGE. You can consider select t-test or Wilkinson for running the test, because Venice is optimized for single cell analytic data and therefore will not suitable for GeoMx data. You can run it that way. Okay, I will go back here quickly. Okay, using either t-test or Wilkinson, and you will have the list of macro gene table in here. You can also export this macro gene, or you can select different macro gene to be queried as a gene. Like this one, you can sort like that, and you can select to query as a separate gene that way. Okay, so that's about macro gene, which is a quick DGE. You can also perform the actual DGE, which you can find on the left bar in here. So run two group is quite the same. However, just remember to avoid Venice for GeoMx data, and that should be fine. Okay, and within the DGE analysis, you can see that all genes, they're included in the DGE analysis by default. However, you can also exclude some of the gene. You can always exclude some of the gene as well. And then we can go next here. Okay. And you can also select the cells or like the ROIs coming from different, different data field or like different samples. Like in here, I select the same GFAP, however, coming from the sample number 3 versus coming from the sample number 1. Okay, I can go back here to view the result, the differential expression. Okay, like that, and I can view the results in here, and you will see I have like a DGE genes with the the distribution plot and the volcano plot in here. Okay, let me go back here. Okay, now because our time is very short, I will also include like the last thing here is that beside visualizing the genes directly on the image like this one. You can also go to charts and graph. Let me go to the marker gene. You can go to charts and graph, go to visualization, and visualize them using multiple kind of plot, like violin plot, the box plot, or like the bubble heat map. And if you want to modify or like customize the figure further, you can select export to BioVinci. So what can you do in BioVinci? Now here, like I have an example in BioVinci. One of the um, very common command that we receive is they want the legend and the text to be bigger. So when you export to BioVinci, you can easily do that. Let's say that for the Fox X one here, we can make it bigger. Okay. And I can also make the legend bigger. And another common request is to readjust the order of different groups. So in order to do that, you can go to advanced feature in here where we have the data order feature. So I can see that the nature of union is quite high. So you can actually simple like drop and drag the nature of union upward like that. And then you can just manually change the order of different groups so that it can represent your data in a better way. And at the end of the day, you can do that for multiple kind of figures, and you can also save and finish and save this figure for later use. Okay. Yes. And that's also everything. Sum up the presentation for GeoMx data today. So it's like a quick tutorial actually, ranging from data submission, pre-processing the data, processing the data, and also some type of downstream analysis that you can perform. So please let me know if you have any question or like, any other thing that you would want to know more about the GeoMx data. Feel free to put in the Q&A. We also put like a quick pause in here. So I will do a quick pause in the screen so that you can, please give us some of the feedback so that we can do, better in the upcoming SCSE Club. Yeah. Okay. Okay. So I will give you guys like about 1 minute to fill in the poll and also to put in the Q&A. Oh, let me see. Compare. Oh, it is possible. Oh, we have one question from Showtime here. It is possible to compare multiple metadata combination in DGE, cluster 1 of sample A versus cluster 1 of sample B. Oh, yes, you can do that. So there are two ways to do that actually. So let me go back to SpatialX. The most simple way that I would say to go with that you can combine the metadata, right? In the metadata field in here, we have the little, I should wait for it to load, okay? There is a manage metadata button where you can add new metadata to combine different metadata together. Like let's say that this can be sample and cluster, right? You can combine multi-metadata, and I can put the batch information in here. I can also put the Louvain cluster in here and save it as a new metadata. Okay, and I can find it here. Let me find sample and cluster like this. So you have a metadata for this. And now you can go to DGE, run new one, and you can select the corresponding group from the corresponding metadata. Okay, sample and cluster. And in here, sample and cluster, and this one. Okay, so this one is sample 1, cluster 0. I can put it in, and now I can go to maybe sample 2. Okay, sample 2, cluster 0. Yes, I can do it this way. So this is one way that is kind of versatile. You can combine metadata in different way to perform DGE. Another way that you can do cluster 1 of sample A versus cluster 1 of sample 2B is to select different data in here. Data here refer to different samples, right? In here, I can have like, this is sample 1. Okay. And down here, I have the sample 2. Okay. And then I can select the Leuven custom metadata of sample 1 and the Leuven custom metadata of sample 2. And just like that, I can select the, the cluster of different samples all together. Like that. This is how you can answer that question. Yeah. So I hope that answers your question, Shota. Please let me know if you have any other question. Okay. Okay. Oh, I think we're a little bit out of time. So that's everything for the SCSE Club today. Again, thank you so much for joining the club today, and we're looking forward to meet you again in the next weeks, the weeks after. I'm sorry. Yeah. Thank you. Bye.